So today it is my great pleasure to introduce our speaker, Dr. Zhengwu Zhang. So Zhengwu is an associate professor of statistics and operations research at UNC Chapel Hill. His research focuses on developing statistical and computational methods for analyzing complex biomedical data with a focus on brain imaging and connectomics. With his background in also computer science, so Zheng will also develop many useful data pre-processing and analysis tools available for the community to use. And then Zheng will receive many honors and awards during his early career phase. So without further ado, so Zheng, I will hand it to you. But for audience, so if you have any question, please type your question in chat during the presentation, and then we should also have some time at the end of the presentation to for questions as well. So Zheng will please take it over.
Okay, sure. Can you see my screen?
Yes.
Okay, great. Thank you, Yi, for the very nice introduction, and thank you, the committee, Hong-Tu, Pan, and God, and everyone else for organizing this wonderful series of seminars. I have learned a lot from this series of seminars, and I'm very glad to contribute to it too. So today I'm going to focus on diffusion-weighted imaging. I'll talk about common ways of analyzing the diffusion-weighted imaging, and then how to optimize your diffusion-weighted imaging, and how to process the data, available pipelines to process this data type. And then I'll discuss some new exciting directions from my point of view. So let's start with the introduction. In our brain, we have two, basically speaking, we have two type of structures. One is called white matter, another one is called gray matter. And diffusion-weighted imaging mainly studies brain white matter and its structure. You can consider the gray matter as CPUs, the computing unit of our brain, and the white matter are those fiber bundles connecting different CPUs. This is one knowledge where you have in a high performance computing center, you have many computer nodes, and there are cables connecting these different computer nodes. And those cables are white matter. So diffusion weighted imaging studies white matter and those fiber bundles. But those fibers or axons are really small, so it's difficult to directly image them. And diffusion-weighted imaging provides a method to indirectly image those fiber bundles. So diffusion-weighted imaging, of course, the keywords is diffusion, and diffusion performs very differently in different tissues. In this case, you can see that we have two different tissues, and the ink is diffused differently. One is isotropically, and another one is anisotropically. It's the same happening in the brain. In some regions, the water molecular diffuse isotropically. For example, in the gray matter, the water diffuse without any restriction. But in the white matter, because of the white matter fiber bundle, they're myelinated, the diffusion is restricted. They're mainly along the white matter fiber bundles direction. So diffusion-weighted imaging is designed to measure the water diffusivity and therefore to indirectly provide orientation information for those fiber bundles. So how to measure diffusion-weighted imaging? Nowadays, we still use a very old design from 1960s, which has been around 60 years old. So by Statistical and Turner, basically you have two diffusion gradients. These two diffusion gradients can acting as stamps. It stamps those water molecular. If water stays still, those stamps cancel with each other, the two stamps, you're gonna have a strong signal. But if water moved and stem does not cancel with each other, you're going to have weaker signal. So the main idea is that if the water has more movement along certain direction, there will be more signal loss and then strong diffusion effect. So we basically have this formula to describe the observed signal along certain direction. It equal to some constant, which is no diffusion at all, times e to the power of minus b times d, where the b is called b value. In this field, it's related to those diffusion gradient. So for example, the duration, the height of the diffusion gradient, and the interval time between these two diffusion gradient. So we often have B equal to 1,000, 2,000, 3,000, etc. in our regular diffusion weighted imaging data. And this D is diffusion coefficient. The bigger of this diffusion coefficient, the stronger diffusion effect and the more signal loss or smaller of the signal. Um, so this illustrates how we're measuring, we're not measuring along one direction, we're measuring along many directions. So each dot represents one direction. And we're also measuring along different B values. So at each voxel, we basically have a 4D function, a function on a four-dimensional space. P represents the directions we're measuring, and B representing the B value we use to measure the diffusion signal. Um, so in Human Connectome project, one of the most famous landmark project, people measure along 270 directions. You basically have 270 dots here. So it's quite comprehensive data. There are key factors to consider in this acquisition process, which we're going to talk about how to optimize those parameters. First is image resolution, the size of the voxel. With high resolution, you have better fiber bundle resolution, but because the size of the voxel is small, the signal to noise ratio will reduce. So it's always a trade-off. And there are also a number of diffusion directions. And the more you measure, the easier you can estimate this function. But of course, it takes a longer time to measure, and then people will move in the scanner, and therefore you're going to have a lot of noises and inaccurate measurements. So it makes an accurate estimation of this function difficult. And of course, speed is another key to consider. There are a lot of acceleration methods to consider, but those acceleration sometimes sacrifice the accuracy too, which there are always lots of trade-offs we want to consider. And that's why we have a section talking about how to optimize those parameters in this acquisition. So once you obtain this data at each voxel, you can estimate a lot of functions, which we're going to talk about later too. In this case, I give you two examples. One is called tensor, diffusion tensor. You can estimate tensors if you plot them look like this. This indicating that water is mainly diffused along this direction. But the tensor has a limited resolution. We can estimate a function on the sphere. This is called orientation distribution function, describing crossing fibers. This is one direction of the fiber, another direction of the fiber. So with those local information, and you can estimate longer fiber bundles and to describe how our different remote brain regions get connected, and then we can analyze brain connectivities. So those kind of the outline of how we're going to analyze this image modality. So why studying diffusion weighted imaging? I think the answer is quite apparent on this slides because it describes the wiring diagram in the brain, and it's those acting as information highways. And you can apparently see any disruption of those highways gonna cause significant function change of the brain. So it's a gateway to understand brain function and dysfunction, the integrity of this structure, and how they're related to neurodevelopmental disorders and neurodegeneration disease and so on are key factors for us to understand the disease progression and to diagnose those diseases and so on. And of course, it's also very reliable biomarker. So this figure basically showing that if you increase the resolution of the structure connectivity, the identifiability increased significantly. This is a paper from Manso et al. From at 2021. And by compare with FC, you can see that FC, the reproducibility is a lot lower, and you cannot increase the reproducibility or identifiability by increasing the resolution. But for structure connectivity, it's much, much higher. And it's also very heritable, which means it has strong relationship to genetic variations. For example, Hong-Tu published this landmark papers in Science a couple of years ago to study the relationship between genetic variations and white matter microstructure. And in one of his figures, he showed that the heritability of those white matter architecture derived biomarkers can have up to 70% of heritability. So it's a reliable, it's a heritable related genetics, and we can confidently obtain this biomarker. That's, I think, give us sufficient reason to analyze this image modality. And next, I want to introduce some common ways of analyzing diffusion-weighted imaging. This include microstructure analysis and then connectome analysis. Okay, so I'm gonna introduce three major different ways of analyzing them. First, of course, is to estimate the diffusion signal and to model the signal. Okay, there are many methods and models for the signal. For example, diffusion tensor imaging and multi-compartment models, which is called NAUTI model. And the second set of analysis include the macrostructure analysis. We try to analyze features or matrix we extracted from the first set of models and to see how they are different across different groups. Those examples include fractional anisotropy, FA, mean diffusivity, MD, AD, RD, and those NODI matrix. And finally, of course, we're interested in the connectome point of view, how different regions get connected and their graph structure, how to analyze those graphs across different populations and so on. So we start from the simplest way, tensor way of analyzing. There are some assumptions if we want to use this formula to estimate tensor. This SK represents the case observed imaging along the GK gradient direction. So D is a diffusion tensor. You can consider it as the inverse of the covariance matrix of the signal you have observed. So you can plot this tensor, and along this longer direction, it indicates water are diffused more. If you recall that the signal is going to be weaker along this direction because water diffuse more. So this type of technique is proposed by Basil et al. In 1994. It's a very widely accepted method to analyze diffusion-weighted imaging. And of course, they assume the diffusion of the water is approximated by Gaussian, which is a big limitation of this method. We'll talk about it later. And from the diffusion tensors, once you estimate, it's very easy to estimate. Only need 6 different directions to estimate it. So you can derive a lot of scalars or features or macro structure integrated measurements. One example is FA. So FA is increasing along this direction. It's also increasing along this direction. The biggest value you can get is 1. The smallest value you can get is 0. And you can also get mean diffusivity from those tensors and AD and RD. Okay, so this is another image illustrated this tensor field, which is a screenshot from this brain region. You can see that the water is diffusing along this direction, meaning that there's this is a very anisotropy region, and there's a strong restriction here for the water to move, and by a larger chance, there's a fiber bundle here. So once we extract those DTI scalars, we can analyze those scalars. We can do regional analysis via some templates. You can have an atlas first about the white matter and then project the atlas to individual subject space. And then for each puzzle, we can extract some summary statistics, say the mean, and then we can extract this from many subjects. We can analyze them. So this is the simplest way of analyzing DTI scalars, FA values, and it's very robust to misalignment because you basically aggregate numbers in this bigger region. Okay, so but of course you're gonna lose a lot of detailed information by aggregating or averaging data in this bigger region. The second set analysis is called TBSS. It's analyze such data at the voxel level, voxel wise level, and this method was proposed by Tom Nichols from University of Oxford, and I think by today this is still the highly cited, like mostly cited paper by Professor Thomas Nichols. What you do is first you determine a study-specific white matter skeleton atlas, and then you project the FA values onto this skeleton, and then you do voxel-wise analysis on the skeleton. It's okay with misalignment because this is a study specific, and then you're analyzing, you're projecting data to the skeleton. It's relatively robust and it's very good for hypothesis generation. So for any clinical study, this is one of the best methods to try first to study whether you have any white matter microstructure difference across the population you study. The third type of analysis is to analyze diffusivity profiles along fiber tracks. We can segment those fiber tracks into bundles, and then we can extract those diffusivity values along those bundles, and then you get these profiles of functional data. And then we can analyze these profiles as functional data analysis. And Hong2 has a lot of this work. He has functional analysis of diffusion tensor track statistics software, which is I think was developed by MATLAB. And I was fortunate to collaborate with him on this quantile functional scalar regression for this type of data analysis. We basically try to treat this type of data, functional data, and then we develop a quantile functional scalar regression to see how clinical variables such as gender, age, and disease status gonna impact the diffusivity profile along some fiber track. Of course, there are limitations of DTI. In this figure, you can see that because the limited resolution of diffusion tensor, there are many different type of structures or configurations of the white matter fiber bundle can correspond to the same tensor, which means that DTI has limited resolution to delineate the underlying white matter architecture. So in order to overcome this limitation, a lot of new models have been proposed. Among them, one of the most popular one is a multi-compartment model called NODI, Nutrient Orientation Dispersion and Density Imaging. It models the observed signal, diffusion signal, as a linear combination of three different signals come from three different compartments. One is called free water, another one is the water inside intranurate, and another one is called extranurate. So this is a mixture model, and you have to estimate a coefficient corresponding to each compartment. So this has been widely used recently, and it requires multi-shell data to fit, which means you have to have multiple B values in your data in order to apply to this model. Once you estimate those compartment, their coefficient, and you can obtain some ratios, free water ratio, NDI and ODI to delineate the underlying white matter architecture characteristics, and then you can analyze those similarly to the FA scalars too. Okay, and of course you have more assumptions here, and the model is more complex. we have the risk of overfitting, or we could have the problem of a convergence, meaning that those coefficient is going to be not be accurately estimated. So it has to be used with care in modeling your data. There are many more multi-compartment models, ball and stick model, Excalibur, and ChairMat, and so on. But those are less popular compared with the Noddy model. And recently, we developed a non-parametric model to just estimate the raw diffusion function at each voxel V. Just want to do it quickly and accurately so that you can resample and harmonize the acquisition protocol and so on. And that's a paper up here already. If you're interested, you can take a look. So those are summarizing the diffusion modeling and how you analyze the features you extracted from those diffusion models. Another big chunk of literature is doing connectome analysis, okay, to analyze the underlying white matter architecture from the connectivity point of view. Of course, this first step is to estimate a white matter fiber bundles. This process is called tractography, and it requires seed points and some arbitrary stopping criterion and so on. And there are many methods that's been published during the past decade for reconstructing those fiber tracks. And all of those methods have serious problems. And Professor Maxim Deskotex, who is a professor in Shortbreak, has organized a competition, and they summarize the results, and they publish the results in Nature Communication. This is one of the mostly cited paper for Maxim by pointing out the challenges, the problems in tractography results. Basically, in the paper, they say that the average precision at the bundle level was 23%. Not mentioned to at the individual fiber level. So the accuracy is very low. Basically, you get a lot of invalid bundles. So that's another problem we're facing. We're going to get a lot of errors in this process. So that should be keep in mind in analyzing such data. And then, keeping that in mind, there's still valid data to analysis. You can see that it carries a lot of information. So people are still improving that. Traditional way of analyzing once you have those tractography result is you parcellate the brain. We parcellate the brain into different regions, and we look at a pair of brain region, how they get connected, and we summarize the connection into one number. We get this connectivity matrix. In this matrix, you throw away a lot of information. On the left hand, it's a really complicated object. On the right hand is just a simple 2D matrix with a dimension, say 68 times 68 or 300 times 300, depending on the parcellation you used to parcellate the brain. So our first work in this field is to develop a more comprehensive connectome mapping framework called PSC. It try to preserve more information. We basically extract connectome information at the streamline level and at the weighted connectivity level by summarizing the connectomes using different numbers, and also threshold them to get a binary network so that we can analyze the brain through the graph the literatures, the network literatures. So at the network level, weighted network level, we extract a lot of different features, diffusion related feature, geometry related feature, for example, number of clusters, average fiber lengths, average deviation from the mean fiber, and so on. We also studied the distribution of the endpoint at the end of our eyes and see extract some properties. So through those, you can extract a lot of different networks, and you start those networks together, you get tensors. Okay, so a more comprehensive way of describing each connection. Instead of using one number, we use a set of numbers. And then we start to analyze those data from simple to more complex at the streamline level, which is, we care about the shapes, the distributions of those streamlines. We analyze such data very differently and publish a set of papers. So it's really fun process. And I want to point out two really interesting way we're analyzing, which we do this a lot, more nonlinear way of analyzing such data. The first case study we do is called the graph autoencoder model, where we use the variational autoencoder framework to learn low-dimensional embeddings of brain networks and to learn a generative model to generate brain networks and then to relate brain networks to human traits, behavior data Y. So this gate contains two modules. One is called inference network, another one is a generative network. The inference network has an encoder to compress our data into a low-dimensional vector Z, which follows a Gaussian distribution. And the generative network basically decodes the Z into latent positions in some latent space. And from those latent positions, we can recover the graph A back. And from Z, we can also predict the behavior traits, the cognitive ability, the substance use, and so on. And the most interesting behavior of this model is allow us to generate synthesized data. So given Y are the cognitive measurements, we can generate different Ys, we can generate different graphs, and we can learn that what the model is learning from our data. In this example, when Y is different, Y equal to 60 or 138, this is measuring the reading ability in the reading test. You can see that between the left and right hemisphere, for different Ys, you have different connections. The connections are increasing. So this allows you to inspect how Y is impacting your brain connectivity in this population. And in the second case study, we care about some confounds that are going to impact our analysis. These confoundings can be the scanners you use to obtain the data, or for example, the head motion, which we don't really want. The head motion going to really destroy your data, lower the quality of your data, therefore give you different connectome estimations. And FSL has tools to correct for the head motion, but it's not going to work so well. It's going to correct for a parent head motion, but there's still a lot of residuals inside. And even after correction here, we can demonstrate that for different connections, and using the ABCD data, ABCD is measuring the kids' data. We have large motion minus smaller motion, and there's a systematic difference in the connectomes we estimate, indicating that after we eliminated the motion using FSS toolbox, the motion is still a confounder to impact our connectome data. We should consider that in our analysis. So we employ the framework we developed. We want to map the brain network into a motion-free version. So how to do that? We first build an encoder to lower low-dimensional embeddings or low-dimensional representations of our graph. We call it Z, and this Z should be independent to the motion from the eyes subject. And then we have a decoder. The decoder going to utilize this Z and the motion together to decode the original brain network. And then we also want the Z to achieve another property, that is to be able to predict the trait in a motion invariant way. Okay, so because we have learned a decoder that can decode both the lower dimensional representation of the graph and the motion, we can arbitrarily set the motion to 0. We can just purposely set the motion to 0. Therefore, we get an A star, which is motion adjusted brain network. It's like a data harmonization or a denoised function of the denoised graph from our data where we learned. So ever since the parameterized using neural network, the encoder, decoder, and the regressor, and our goal then is to optimize this neural network to achieve a good fit of our data. And this illustrating this the result, the most interesting results. In the ABCD data, we extract the large motion, small motion networks, and we plot their two PCs. We do PCA and then plot the two PCs. You can see a clear separation between these two populations. But after you denoise the motion, once you plot the PCs, you don't really tell the difference between these two populations. So we then can conduct analysis in motion invariant way. So this is our two illustrations we have done two illustrations for what we have done for discrete brain network analysis. And nowadays we don't do a lot of discrete brain networks analysis. I want to introduce another set of work we've done recently to address limitations for discrete brain networks. So we have so many different postulations. Each of them are going to give us a different brain network. How to choose from those postulations? We at least has 100 of them now. With one postulation, you get one set of results. With another postulation, you probably get a different set of results. So it makes the reproducibility of your study very difficult. And also, there are huge of information loss. Once you use a postulation, the sub-postul level connectivity information is a disappear because you don't consider within brain within postul connectivity. If you're interested in more details on this, there are papers here. For example, we have a correspondence in Nature Reviewer Neuroscience to talk about issues of the postulation in structure functional coupling, which rely on the connecting computed from structure and functional connectivities. So the solution to address those limitations is to treat the connectome as a continuous functions, So let me explain that. What do I mean to treat them as continuous functions? Of course, in order to do that, we have to again develop some data processing pipelines. The first work to do this is called SBCI, surface-based connectome integration, where we want to map all the connectome information, both structure and function, to the cortical surface, and then we can analyze them in a very high resolution manner from the continuous point of view. They became continuous functions on the omega cross omega space. So the omega is actually a union of the two spheres, left and right. From the pipeline we developed, what you obtain is actually those fiber bundles, and then those fiber bundles, each of those fibers, they're connecting the cortical surface. Cortical surfaces are complex manifold. We actually get the endpoints. We care about the connection. We don't care about the actually shape for this moment of those connections. So we get a pair of points from each fiber bundle connecting different cortical surfaces. We're going to get a lot of those pairs. For example, 500,000, 1 million, or 2 million of those pair points. Those pair points describes how different brain regions get connected. So that's the data we're analyzing now. And if you inflate the cortical surface into a sphere, you can get this points on the spheres too. Sphere has simplified geometry compared with the original cortical surface. So many times we're going to analyze those points, the connectivity on the spheres. Okay, so for one object, we're gonna model this pair points observed as a stochastic point process on omega cross omega. Omega is the cortical surface, omega cross omega because we have two points. And for any two brain regions, E1 and E2, the number of fibers connecting them equal to the integration of this intensity function. So this is called continuous function. The continuous structure connectivity, the earliest concept was proposed by Measure et al. In their 2017 paper and measures from Paul Thompson's group. And if we normalize this U, we're gonna get a PDF. And if you have multiple subjects, and then you can have this generated model. So you first sample a U intensity function from some random process, and you sample some integer from Q, indicating how many fibers you have, and then you can sample this O's, the point clouds. And then with this data object, we have a few research problems, at least, something we're dealing with. First, we need to estimate this U on this omega cross omega. It's a high dimensional density estimation problem on a nonlinear manifold space. So it's a relatively new problem, and then we can use kernel-based methods, and we face two challenges for how to define a kernel on the manifold and how to select the bandwidth and how those two selections gonna impact your downstream analysis. If you're interested in this type of work, you can look at our new paper in imaging neuroscience. And the second type of method is to using GP to estimate this U, but the GP is problematic here because the domain is a six-dimensional space and GP scales cubically with the number of data points in this domain. So in order to estimate high-res connectons, we face computational challenges, and we have a paper to address that. It's quite interesting. I think it's gonna appear very soon. Another really interesting work is to use neural fields to represent this complex intensity function. So the function becomes a neural network, and for any location, you input that location into this neural network, you're gonna get the value out. Okay, so this is really interesting field. I learned a lot through this line of work. So it's related to the representation power of your neural network and how you can specify those activation functions and the convergence and so on. So it's very interesting work. It's a bridge the theoretical research of neural network with the actual application of those neural networks too. So I encourage everyone to pay attention to this set of literature. It's a growing field too for analyzing functional data. And the second set of problem, so once you estimate this U, you have to align them across subject, right? Because different subjects has different cortical surfaces, and alignment is very important. And our goal is to find a deformation of those nonlinear manifolds so that we can align connectivities. Those connectivities are continuous functions. Okay, so this I think is also very interesting work. We have an archive paper for this type of research where we're going to use connectivity information to align the brain cortical surface. I think it's a really interesting contribution to the cortical surface alignment set of literature. And the third set of analysis is to link U to behavior traits, to do trait prediction, disease prediction, to do global hypothesis testing, or identify local subnet differences between groups. And when U is estimated, and it's a semi-symmetric functional data analysis type of problem. When U is not estimated, you have those point process on manifold, and you need to develop methods to analyze a population of those point processes, which I don't see a lot of paper in the literature, and this is quite exciting work to do. Also, we're doing a lot of this type of analysis recently. Okay, that's basically summarize popular ways for analyzing diffusion weighted imaging. And of course, I want to borrow a figure from Professor Bing Yu's PNAS paper called the Vertical Data Science. She has this data science life cycle. We have done a lot of this data cleaning, modeling, and interpolation. As a statistician, I think we also need to contribute to the data collection point view step. As I pointed out earlier in this talk, that DTI, you have to consider a lot of things. You have to consider the spatial resolution, the number of directions to get, the acceleration, and we need to understand how each choice is going to impact the downstream statistical analysis. Predictive model, how to tell the difference between two populations and so on. The very less amount of work in the literature, And I'm going to show you a couple examples that we have done to contribute to the data collection. The first one is the popular way of sampling diffusion weighted imaging is to uniformly sample along different directions. So you have a sphere, you just uniformly put points on the sphere, and those points give you the directions you want to sample. But we want to ask the question, like, do we need to sample this many, like 90 directions per shell? Or should we use 60? Should we use 40? What's the best number to use? And this is not well known in the literature. You can see that ABCD, Adani, UK Biobank, each of them have their own experts and they just suggest different numbers. And they don't, there is no consensus on how many directions to sample and which shell. So in order to handle this problem, we have this paper, Optimized Diffusion Imaging for Brain Structure Connective Analysis to study the optimal way to sample the directions so that we can best estimate the information we want from the data. The second set of research we do is to see how acceleration, multi-slice, multi-band methods gonna impact the downstream analysis, connectome analysis, macrostructure analysis, how implant acceleration gonna impact those analyses. So we have found that motion decreases significantly with acceleration because with acceleration, you're going to reduce the acquisition time and head motion will be reduced. But signal to noise ratio will drop significantly at high resolution. So you have to consider that. Don't accelerate too much. And diffusion matrices are biased by high resolution. And so basically with different acceleration, you're measuring different things. It's really important to harmonize your data. If you don't harmonize, you basically compares oranges with apples and so on. So after analyzing those, we have recommendations. For example, you have to avoid implant acceleration. It was really popular in the literature. You see the data, you see people still use implant acceleration. And also, don't accelerate too much. Multi-band 3 achieve balance of speed and reproducibility. And this paper is to appear in Human Brain Mapping, so we should have a follow-up paper on this too. And finally, if you want to jump into this field, we have seen a lot of different ways of analyzing. Is it very easy to jump into this field? I think now it's much easier compared with 10 years ago. For pre-processing, first, we have seen that diffusion weighted imaging is very noisy. The signal is kind of weak and artifact rich. So QC is strictly needed. You have to inspect the head motion. You have to inspect bad data frames. You try to replace those and reject really bad data from your analysis. Otherwise, your analysis will be biased by those outliers. And we have to correct for many artifacts, or the methods to do that, head motion, eddy currents, and suspectabilities. After you've done that, do connectivity quality control and do tractographic quality control too, so that you can have clean data to analyze. And eddy currents is caused by unwanted magnetic fields, and those are gonna distort your data. And this is because you're gonna rapidly switching of the gradient, positive and negative gradient and so on. All of this is gonna cause unwanted magnetic fields in the tissue you're collecting. So correct the artifacts is very important. And there are also geometric distortions due to air tissue interface. So we have to correct this. Also, in order to correct these artifacts, we need to acquire AP and PA image. And in many studies, people just collect just one direction instead of both directions, and making the correction very difficult, and therefore the analysis very inaccurate. So from the data collection point of view, like collected the right amount of data for downstream analysis is also very, very, very important. And then processing tools. We have traditional tools. We have FSL, which is really good for eddy current, susceptibility correction. And we have M-matrix, which is very good at fiber orientation estimation, spherical deconvolution, and tractography. We have DiPy. This is maintained by Maxine and a lot of his collaborators. It's a pure Python based. It's a package you can install and it's really easy to use. And DIS Studio, it's GUI based, very user friendly. This is maintained by Frank Yer from Pittsburgh. Traditional tools sometimes has steep learning curve. You have to, combine different tools together. We talk about a lot of artifacts and so on. You have to correct them. You need to understand the steps to correct them, the order to correct them. So it's a very complex to use. But we have Warper tools now to simplify this process. DMI prop, QSI prop, or the surface-based connected integration from our lab, which can help you to analyze both FMR and diffusion MR at the same time and output the surface-based connectome, which you can do very detailed connectome analysis. And all of those tools are BERTS compatible. You can format your data into BIDS format and then just call sometimes just one single line and you can finish your analysis in a couple hours or a couple days, depending on how many steps you're running. And then most importantly, I think for all of those, I believe they have Docker, at least we have Docker and Singularity or app container containers images. You can just download this image, so which meaning you don't need to configure any software into your local computer. Just download the image and then run the command and from the container, and you will be able to analyze your data. So a lot of tools now available at very convenient level for this type of analysis. And finally, I want to talk about exciting directions. To name a few, I think first is validation of the connectome. Their new development, I think, is very exciting. For example, polarized light imaging, which allow us to accurately image the architecture use at very high resolution, and this allow us to validate the data we got from the estimate we got from diffusion weighted imaging and their tracer study, his logic study too, a combination of those together to validate and improve our tractography algorithm is very exciting direction. And of course, combined with other modalities, combine diffusion with FMR so that we can link structure and function network together, and diffusion plus QMR so that we can link macrostructure plus tissue composition together, diffusion plus PET, we can jointly model structure and then molecular properties together. So how, tau and beta, those amyloids are propagating with the development of Alzheimer's disease and so on. And of course, I would like to promote our atlas-free connectomics. You don't need to consider an atlas. You're analyzing the connectome at the super high resolution. A lot of new methods is needed to analyze such complex data. It's a very interesting field. And finally, AI and model-free approaches. Deep learning is very good at learning the patterns in the data, so we can speed up the whole process a lot. Given the images, how to estimate the macrostructure, matrix, do artifact correction, estimate connectomes, and then do data-driven harmonizations. And generative models too, diffusion models, VAEs, they're very good for super resolution, synthetic data generation, and help if you don't have enough data, this model can essentially help you with your inference for very little data. That's another very exciting directions. Finally, I'd like to acknowledge my collaborators from many different places. I was lucky to work with them for this topic and funding support from NIH and other places to support our work. And finally, I borrowed a few slices from Martin Steiner, he's another expert in DTI at UNC. So that's all for my talk. Thank you.
Yeah, thank you, Jung. Really great talk. I think there was a question in chat. So, Kazim, do you want to unmute yourself and then answer the question?
Okay. So the question is connectivity matrix a square matrix? What does
Yeah,
each
thank you.
describe? Or is the matrix generated from two brain as an example? It's a square matrix. It's each element describes the connectivity profile between two brain regions. So you consider that as a adjacency matrix from network data. Okay, that's the data you see.
Thanks.
Yeah. Hi. Nagra.
Yes. Yeah, Nagra.
Ah, thank you. Thank you. Thank you for a wonderful talk. My quick question is, which of these methods is being used in clinical practice right now? And which might be the next one that might be used in the next, say, few years, perhaps?
So there's echo. I just find it's difficult to understand your question. You're asking diffusion weighted imaging has been widely used in clinical, or is it going to be used in clinical study?
Yeah, which method is being used right now in clinical practice?
In clinical practice. So, from my observation, we see a lot of more researchers or studies are collecting those diffusion weighted imaging. It was not so popular many years ago. If you look at clinical research like Alzheimer's disease ADNI data set, you can see that now they put more emphasis on diffusion weighted imaging by using more advanced imaging acquisition techniques. However, because they just start to collect such a data, it's very limited. There's very limited amount of data for us to understand. With the current fancy methods, you see multi-compartment models, those high-res connectivities. So we still in clinical data, we need to implement those advanced acquisition methods to collect a large amount of data and then start to study them and understand the value of this image modality as a biomarker. So there's still a lot of things to do in order to develop some reproducible, credible biomarkers for clinical studies. I see more and more, but we don't really have, a real good applications that has been, let's say, FDA approved for our clinical research yet. Yeah.
And Jasper?
Hello. Thank you
Hi.
for the wonderful talk. I'm Jasper. I'm a PhD student with Rogier Mars's group at the University of Oxford, and I've been doing tractography for my PhD. My question is, what do you think about more traditional machine learning approaches like clustering and dimensionality reduction, which are a bit shy of the generative AI approaches that you're describing, and the variational autoencoders? What do you think about this middle ground where it's machine learning but not quite deep learning or anything of that nature?
Yeah, I think those classical machine learning methods are still very useful, especially when we don't have ground truths, right? This supervised learning can help us to clean the data. At least you can filter the apparent outliers and so on, which can be very important in your analysis, right? Once you have ground truths, those machine learning based, deep learning based methods can help you to learn those ground truths very quickly. But the issue with DTI is we don't really have ground truths. But still, I mean, those deep learning methods, they are very good at learning. So which means, if anything in your data, bad or good, they can learn it. You have to be able to distinguish what kind of signal you want the deep learning method to learn, what one you don't want to learn. One example is the motion one I gave, right? So you want to learn valuable information, but you want the learned representation to be invariant to some factors that you don't want. So I think there's still like a lot of really good applications for those deep learning generative models, but we have to use them very carefully and make sure we know what it has learned, whether it's going to be valuable for our downstream analysis for clinical research. In general, I found those deep learning methods, we have to use them very carefully. The representation we learned, it can be contaminated by, factors such as scanner impacts or motion impacts and so on, which we don't really want in our data. Yeah.
Wonderful. Thank you.
Thank you.
Is there any other question for Zhengwu? If not, so just to give you a heads up for our next seminar. Oh, sorry, I saw one in chat. Oh, not the question. So our next seminar will be October 8th from 2 to 3 from Dr. Russell Pajak. So he's the Albert Royline Professor of Psychology from Stanford University. He's also the main driver of the reproducible neuroscience. And he will talk about reproducible neuroscience next time. And then Dr. Pajak is also the first speaker we invited from the domain area from neuroscience. So he will also present another angle in terms of thinking about like analyzing neuroimaging data and then implementing AI tools as well. So please join us next time as well, and hope to see you next time. And then thank you, Zheng, again for your wonderful talk. And then thank you all for coming.
Yeah, thank you all for coming. Yeah.
All right, thanks. Bye, everyone.
Bye.